Hey there, I'm Mark Junius, second year MARC trainee, and I'm here to tell you about all the fun plant research I've been doing this past 2 years. Now, my research focuses on how plants react to abiotic stress, specifically salt. Now, I'm sure we're all aware that too much salt in plants don't exactly go well together. But I'm interested in asking why. What happens at the genomic and molecular level when plants are exposed to too much salt, and how can plants combat this? By far the biggest cause of saline soil is drought. Simply put, less water in the environment means less solute dilution and higher concentrations of salt in the soil. Less water available means farmers have to resort to poorer quality water as well, only exacerbating the problem. Considering the fact that drought has only become more frequent and severe due to climate change, saline soil is a challenge agriculturalists will have to face. It's estimated that by 2050, over 50% of arable land will be impacted by salinization, which is terrible when we need that land to feed our ever-growing population. In the Central Valley of California alone, crop losses were estimated to be over 3.7 billion in 2014. So even if you're not much of a humanitarian, it's bad for business too. Now, I'm sure for the most part, most of us know that too much salt is pretty bad for plants. I'm sure we've all done a demonstration where you put a plant in really salty water and it got super flaccid, but there's actually a lot of things that happen when plants undergo salinity stress. One of the things that I'm interested in is something called ion homeostasis. Now, to explain this, I'm going to do a little bit of a demonstration. Now, when plants like this philodendron right here take up water, they're not just taking up water, they're also taking up a lot of ions in the soil. Now, one of these ions is potassium. So potassium is really important to plants. Potassium is an important cofactor in a lot of processes in plants, including things like ATP production, photosynthesis, and even opening and closing of stomata. Now, because potassium is so important to a lot of metabolic processes in the plants, that means they're going to need a lot of it. But plants actually have a higher concentration gradient of potassium when compared to the soil. So that means they're going to have to resort to active transport to get a lot of it. Let's say this is the outside of the plant over here, and this is the inside of the plant cell on this side. These specific transporters are going to have affinity for potassium, allowing them to move inside the plant, sort of like this. Now, potassium is not the only thing we have in the soil. We also have sodium. So the thing about these two ions is that they're actually relatively similar. Other than the apparent size difference in that potassium is a little bigger, they kind of act the same way. They both have a similar charge and are really close to each other on the periodic table. And this is going to be a problem as those active transporters aren't really going to be able to discriminate between potassium and sodium. So even though these transporters are going to be taking up the potassium that they're supposed to take up, they're also probably going to have to take up sodium as a byproduct just because it's so similar, and it's not going to be able to tell the difference between the two for the most part. And the problem with this is that all those important functions that potassium was helping out in, sodium can't do. So it's just kind of going to go in the plant and just build up and stay there, and that's actually really toxic towards the plant. Now, in normal conditions, this might not be the biggest deal, but when you have excess saline soil, this is going to be a huge deal because that means there's going to be tons of sodium ions, and there's going to just be a rapid fire of them flowing in, and it's going to be going to the point where you're going to have more sodium ions being taken in than potassium ions, which is going to screw up all those enzymatic processes going on in the plant. It's important to note that sodium toxicity can have varying effects depending on where it accumulates in the plant. In the root, where the primary functions are storage and uptake, the plant cells will be more tolerable to excess sodium. However, in the shoot, where primary functions include things like photosynthesis and stomatoregulation that need a lot of potassium ions, the cells are more vulnerable to toxicity from sodium accumulation. Now, salinity has an effect on all plants, but I'm specifically interested in a specific one. Almond. Now, there's a lot of great things about almond. The trees are really pretty, they're healthy and nutritious, and they're also a $21.5 billion industry in California. So it's really important that we maximize the growth of almonds. Not to mention, they taste pretty good too. Now, as good as salted almond tastes, almond trees don't exactly deal that well with saline soil. They're big and they uptake a lot of water, which means they're going to be uptaking a lot of those sodium ions as well. In addition to this, almond trees aren't exactly adapted to grow in high saline environments, so that ionic stress in the shoot can really build up and be harmful to the plant. Now, in addition to all that, almond is kind of unique in that a lot of agriculturalists use a technique called grafting to grow it. Basically, grafting is a technique in which you take the top part of one plant, referred to as the scion, and the bottom part of another plant, referred to as the rootstock, and you sort of put the tissues together, and eventually they'll connect and combine, and you'll get something looks like this, and this is essentially a chimera. Now, what's important about grafting is that different rootstocks have different qualities and are adapted to different conditions. With the right rootstock, He can grow really vigorous almond trees, or even grow almonds in places that may have been unsuitable without a rootstock. Now, the rootstock that I work with is called Nemagard. Now, Nemagard is a really popular rootstock that's actually derived from Prunus persica, or peach. It's commonly grown for its resistance to nematodes, but it actually has two qualities of interest to me. One is that it's been shown to provide better salinity tolerance to almond crops, and another is that Prunus persica has a completely sequenced genome, which is really important to me if I want to understand genetic and molecular mechanisms of this plant in regards to salt tolerance. So, even though there's a genome sequence, we still don't really know anything about how almonds or rootstocks are able to combat salinity tolerance, let alone at the genetic and molecular level. However, there has been some research on one specific gene in our favorite model organism Arabidopsis that caught my eye. This gene is called HKT1, or High Affinity Potassium Transporter 1. In Arabidopsis, HKT1 encodes a plasma membrane sodium uniporter that relocates sodium to areas of low tolerance to high tolerance, or root to shoot. Now, the process of how this works goes a little something like this. So, to understand HKT1, we first need to understand how the vascular tissue of the plant works. First, we'll talk about the xylem. The xylem consists of dead, water-conducting cells that transport water and minerals up the plant. Think of it like a big straw powered by the sun. The phloem moves in the opposite direction. Sugars, ions, and minerals that are moved from the leaves of the plant, or the source, to areas of storage like the roots, or the sink. As water and sodium ions move up through the plant through the xylem, HKT1 transporters take in sodium ions in the nearby xylem parenchyma cells, acting like as a net to prevent accumulation into the shoot. Meanwhile, in the phloem, HKT1 transporters in the phloem companion cells load sodium ions in the phloem that had accumulated in the shoot so that it can be transported away from the leaves and into areas that are more salt tolerant, such as the roots. Now, that was all done in Arabidopsis, but my question is, does HKT1 play a role in salinity tolerance when it comes to almond rootstocks? One of the issues with answering that question is that there aren't a lot of resources or information about almond rootstocks. Not to mention, almonds are big, take forever to grow, and are expensive. However, there is a workaround we can do to test the first question we need to address, which is whether HKT1 is even functionally conserved in these almond rootstocks. We can do this using a functional complementation study with Arabidopsis. Arabidopsis is a model plant organism that has tons of resources, grows easily, and has a rapid lifespan. Essentially, previous studies have shown that expression of the HKT1 found in Arabidopsis, which we'll refer to as AT-HKT1, aids in conferring salt tolerance, and knocking the gene out reduces salt tolerance. However, if we remove it and transgenically insert the rootstock HKT1, which we'll refer to as Prunus persica HKT1 or PP-HKT1, can we restore salinity tolerance? To test this hypothesis, we'd first need to do a couple of things. One, we would need to clone and isolate the rootstock HKT1, and when we do that, we also need to create transgenic Arabidopsis lines that express this rootstock HKT1. Second, we would need to test the effects of salinity stress and compare them to control. How do these transgenic lines fare in terms of salt tolerance, and is this gene being expressed? The Columbia line serves as the wild type and the positive control. Essentially, this is an Arabidopsis plant that is untampered, meaning it has a regular expression of Arabidopsis HKT1, also known as AT-HKT1. Our next line, which we'll denote with the lowercase AT-HKT1, is a mutant line. Essentially, by using a tDNA insertion, we create a knockout mutant at the AT-HKT1 sign, meaning this plant will have no expression of HKT1 whatsoever. Since this line lacks any HKT1 expression, we would hypothesize that it would have reduced salinity tolerance when compared to the Columbia line. Our next line was our first transgenic line, which is the native promoter line, or NP6. Like the mutant, this line has a non-functioning AT-HKT1. However, it does have a transgenic expression of HKT1 in the form of the Prunus persica, or rootstock HKT1, which we'll denote as PP-HKT1. Assuming the HKT1 gene found in the rootstock is conserved, we would expect the line to at the very least have higher salinity tolerance than the mutant line. Last but not least is the overexpression line, which we'll denote with OE2.2. This transgenic line has a similar construct to the native promoter line, but rather than having the natural promoter region found in the HKT1 gene, this line has a modified 2x35S constitutive promoter that induces overexpression. Depending on the method of how HKT1 is expressed, it's possible that overexpression can lead to increased or decreased salinity tolerance. The result can serve as a good indicator of how HKT1 functions. Of course, first we have to generate the transgenic lines. The process goes something a little like this. After selecting tissue from the rootstock, we can isolate RNA using a triazole reagent. Using reverse transcriptase PCR, we generated the cDNA of the rootstock HKT1. The next step was to clone the gene. This was done using gateway cloning technology in E. coli. Gateway cloning is a technique that allows the transfer of DNA fragments of a gene of interest between cloning vectors. It's a bit complicated, but essentially it involves a reaction called a BP reaction to generate an entry vector for gene amplification, and then an LR reaction to create an expression clone with the gene of interest. The DNA can then be purified and extracted and placed into agrobacterium. Agrobacterium is a bacteria with a unique ability to be able to integrate some of its DNA into the genome of a plant. Scientists have modified this bacteria so that it will insert a gene of interest into the plant. In this case, it will be our almond rootstock HKT1. By dipping the flowers of mutants into a sucrose agrobacterium solution, we can generate seeds of our transgenic lines. In addition to the HKT1 gene, our construct also included a hygromycin resistance gene. By placing the seeds on a hygromycin medium, we were able to ensure that only those seeds that were successfully transformed could germinate and grow. This selection process was repeated multiple times until we obtained a T3 generation in order to ensure that a homozygous line was obtained. All of our lines were then screened to ensure proper constructs using genomic PCR with specific primers. From here, we were ready to experiment on our lines. Now, our goal was to conduct multiple salinity tolerance assays and see how the lines fared. All of the plants were germinated on agar plates, and then the seedlings were placed on soil medium and grown with a nutrient solution for 18 days. From here, using a sample size of 12 for each line, we created a control and experimental group, with the control receiving a nutrient solution for another 18 days, and the experimental group receiving a nutrient solution alongside 90 millimolar sodium chloride. Here is a sample of the results. Although there appears to be no discernible difference between the lines with sodium chloride absent, it is apparent that the mutant line didn't fare as well when compared to the transgenic or control lines when sodium chloride was present. When examining the quantitative results of relative dry weight, our results matched the trends of the hypothesized outcome. Relative dry weight was obtained by creating a ratio of the salinity treatment group and the control group for each of the 3 biological replicants. An ANOVA test with p-value less than 0.05 was used to test for the significance. It is very apparent that the 2 transgenic lines had a higher relative dry weight and thus higher salinity tolerance than the mutant. In fact, there was no significant difference between the control line and the transgenic lines as well. Of course, that's only one part of the equation. We also had to show that this salinity tolerance was the result of the Prunus persica HKT1 expression. To do this, we used qualitative reverse transcriptase PCR to measure expression. For both native promoter and overexpressor, it was clear that the addition of the salt treatment induced higher expression of the HKT1 ortholog. Now, an interesting result was found when we continued the salinity treatment for 30 days on the 4 lines and then measured the survival rate. Although the survival rate to the wild type and the native promoter remained high, the overexpression line had a reduced survival rate to around 50%. This suggests that overexpression of the HKT1 gene may not be ideal, and other research has suggested that site-specific expression of HKT1 may be the more ideal way. Now, I know what you might be thinking. Were you blabbering on about sodium and potassium ions? When are you going to start talking about them? That was exactly the next part of my experiment. And to do that, I was going to use a technique called ICP-OES. ICP-OES, or Inductively Coupled Plasma Optical Emission Spectrometry, is a technique that can be used to measure concentrations of ions such as sodium and potassium in millimoles per kilogram of tissue. Now, we had one challenge when it came to doing the ICP-OES. You see, when you do it, you have to make sure that you have really clean tissue, including the roots. But when you have plants that are really small, it's really hard to remove all this soil from the roots, and you're often going to get soil contamination, which is not what we want. So we would have to find a workaround when doing this process so we get clean roots. One way to get around this obstacle was through the use of hydroponics. Hydroponics is a method of growing plants in an aqueous nutrient solution rather than a soil media. That way, the roots would be free of soil contamination for the spectroscopy to work. So things were looking pretty good. I had set up a hydroponics system, was ready to start running experiments, and I also learned that I was going to be spending my summer doing RAU in Cornell, and then a worldwide pandemic hit. It wasn't all bad though. Dr. Corinne LaRoque in her lab offered to teach me bioinformatics over the summer. Here, I was able to learn the computational analysis aspects of RNA-seq as well as other things such as R coding that I could use in my future projects. As of quarantine, I have been applying these bioinformatics skills towards my almond rootstock project. My newest project consists of examining RNA-seq data between two different rootstock lines under salinity stress in search of differentially expressed genes. This summer, I interviewed with 4 different schools for a plant biology PhD program, including UC Riverside, UC Davis, Cornell, and UC Berkeley. It was a tough application cycle, but I'm happy to say that I was offered acceptances to all 4 of them. After I graduate this August, I'll be joining UC Berkeley's Plant and Microbial Biology department. And I'm hoping to do some cool plant research, and I actually kind of want to venture into algae, so we'll see how that goes. Nice jacket. I'd like to thank Dr. Amy Litt and her lab, Dr. Davinder Standu, Beswa Acharya, and Amita Kondal from the USDA. And in addition to this, I'd like to thank Dr. Corinne LaRoque and her lab, as well as a big shout out to Rebecca Brown, Dr. Martinez, and the MARC USTAW program. I hope you all enjoyed seeing what I was up to these past 2 years, and this was really fun making, so thanks for watching.